Welcome to the lecture on specialised circulations. Different organs have different needs at different times. For example, the oxygen demands of the heart and skeletal muscles increases in times of high activity. This requires an increase in blood flow to these tissues, which must be compensated by reduced blood flow elsewhere, because the total volume of the circulation has to remain unchanged. To accommodate the varying needs of different organs, the vascular system has evolved some special features that are characteristic of particular vascular beds. I don't intend to cover all the body's vascular beds in this lecture. Instead, I will focus on a few circulations that have unique or particularly interesting characteristics which are essential for their function and which distinguish them from other parts of the vasculature. So in this lecture, we will explore the circulations of the skin, the heart, the brain, the lungs, and the fetus. Other specialised vascular beds are the splanchnic circulation, which serves the visceral organs, the circulations of the kidney and skeletal muscle, and also the lymphatic vessels. We will begin with the circulation of the skin, known as the cutaneous circulation. The blood vessels in the skin are specialised both in structure and function to help the body lose heat when it is hot and conserve heat when it is cold. In that way, it works to maintain body temperature at a steady 37 degrees centigrade. The dilation of blood vessels in the skin diverts blood from the body's warm core to the cooler surface where it is able to cool down. Constriction of cutaneous vessels keeps blood within the warm body core, preventing heat loss from the skin surface. Blood arriving under the skin in arteries enters the smaller arterioles, which transport the blood through the dermis. From there, the capillaries distribute the blood throughout the epidermis. Blood then leaves the capillaries to enter the venules, from where the veins transport it to the heart for reoxygenation. So far, this process seems like any other circulation, but the cutaneous circulation has an additional mechanism for maximising the distribution of blood to the skin when it is hot. First of all, notice that the venules running through the dermis are actually a plexus or a network of veins. In addition, there is a direct connection between the arteries and the venous plexus, which is known as the arteriovenous anastomoses. In cold temperatures, these anastomoses are closed. But when heat loss is needed, the anastomoses open and a large volume of warm arterial blood can immediately invade the venous plexus, rapidly increasing the amount of blood next to the skin surface. The anastomoses are composed of smooth muscle and can be regulated in the same way as the smooth muscle of the main blood vessels. This slide summarises how the cutaneous circulation functions. The main regulator of skin blood flow is the sympathetic nervous system, the outflow of which depends on the central nervous system detecting signals from temperature receptors in the skin, as well as the core body temperature detected in the hypothalamus. The skin arterioles and anastomoses are particularly sensitive to noradrenaline, which can constrict them to the point of obliteration. In extreme cold, the prolonged obliteration of these vessels in the extremities can lead to frostbite and eventually tissue necrosis and gangrene. Skin blood flow can also be regulated by local mediators. Examples include histamine and bradykinin, which cause the redness and swelling associated with inflammation and itchy rashes. For example, when you cut or graze your skin, or have an allergic reaction to something, mast cells in the skin release histamine and trigger the formation of bradykinin, which activate both vasodilation and increased vascular permeability. The coronary circulation describes the blood vessels of the heart muscle or myocardium. The structure of the coronary circulation is illustrated in these figures. The coronary arteries originate at the top of the aorta and transport blood from the aorta along the outside of the heart before branching into the muscle layers. The arteries running deep within the myocardium are referred to as subendocardial. As they are within the heart tissues, the postcapillary veins drain directly into the chambers of the heart. The capillary network in cardiac muscle is particularly dense, reflecting the high oxygen demand of the muscle cells. The consequence is that all the muscle cells are within a distance of about 1 red blood cell from the nearest capillary. This maximizes the delivery of oxygen to the muscle and the removal of metabolic waste. A particular feature of the coronary circulation is that when cardiac muscle contracts during systole, the arteries running through the ventricles become squashed, making it difficult for blood to flow through them. The vessels can become squashed to such an extent that they are completely obliterated. This figure shows how blood flow varies during the cardiac cycle, starting in the middle of diastole when the muscle is relaxed. The aortic pressure at the top of the figure shows the pressure as the blood enters the coronary arteries. This is what pushes blood through the vessels. During systole, the pressure in the aorta rises. This is because the blood has difficulty getting through the squashed arteries, and as it cannot move forward, a back pressure rises. The most dramatic effects on the coronary circulation are seen in the left ventricle because it contains more muscle and contracts more powerfully than the right to eject blood into the systemic circulation. This can be seen in the middle trace. At the onset of systole, as the muscle contracts and occludes the arteries, the flow drops to 0. As systole progresses, there is a small increase in flow as the rising aortic pressure forces it through. But as systole ends and the muscle relaxes, there is a dramatic rise in blood flow as the vessels open up to reinstate flow through the vessel. The consequence is that the majority of the blood flow to cardiac muscle takes place during diastole. A major regulator of coronary blood flow is the sympathetic nervous system, which has different effects depending on the vessel size. The mechanistic basis of this is the nature of the adrenergic receptors present on the muscle cells in the blood vessel walls. When stimulated by noradrenaline or adrenaline, alpha-1 adrenoceptors promote smooth muscle contraction and vessel constriction. In contrast, the stimulation of beta-2 receptors on the muscle cells promotes muscle relaxation and vasodilation. The overall effect of these agents, and hence sympathetic nerve stimulation, depends on the relative numbers of these receptors present. In the large coronary arteries, alpha-1 receptors predominate, so the effect is constriction. This constriction helps to propel blood into the smaller vessels, which are the main determinants of tissue blood flow. The small coronary arteries mostly express beta-2 receptors, so they dilate in response to sympathetic nerve stimulation, thereby increasing blood flow in the tissue and increasing the delivery of oxygen to the cardiac muscle. As cardiac muscle is a highly metabolic tissue, not surprisingly, metabolic factors are important regulators of coronary blood flow as well. As oxygen consumption increases due to increased cardiac work, blood flow increases in order to match blood flow to oxygen demand. A major metabolite in the heart is adenosine, which acts on adenosine receptors in the smooth muscle cells of the arteries to evoke muscle relaxation. The receptors involved are the A2A and A2B subtypes. When activated, these receptors open ATP-sensitive potassium channels in the muscle cell membrane. This causes the membrane to hyperpolarize, which in turn closes voltage-gated calcium channels, therefore limiting calcium entry into the muscle cells. Other metabolites, such as lactate, can also influence vascular muscle and contribute to linking blood flow to metabolism. In addition to these mechanisms, healthy coronary arteries are also capable of autoregulation. This is a mechanism that maintains coronary blood flow at levels appropriate to the needs of the heart muscle and irrespective of the systemic blood pressure. Autoregulation is also a feature of the cerebral and renal circulations, so I will cover it in detail a little later. The cerebral circulation is highly constrained by its environment. Unlike circulations elsewhere, the cerebral blood vessels are trapped within the bony cranium. This means that there is little room for blood volume to change. So blood flow to areas within the brain must be regulated to match oxygen supply to demand, without an overall change in the volume of blood entering or leaving the brain. This means that the blood flowing into the brain via the arteries must be matched by blood leaving the brain in the veins. The sympathetic nervous system plays only a small role in regulating cerebral blood flow. The main regulators are local mechanisms within the brain, although the systemic blood pressure can influence cerebral blood flow. If the systemic blood pressure were to undergo a large fall, this would diminish the blood flow to the brain. An example of this can be seen in postural hypotension. Upon standing up suddenly from a lying position, blood can pool in the leg veins and systemic pressure fall, resulting in a loss of cerebral blood flow, which is experienced as dizziness and/or fainting. This is normally prevented by the baroreceptors, which immediately detect the fall in blood pressure and increase sympathetic drive to compensate. Postural hypotension is common, however, when the baroreceptor sympathetic nervous system pathway is interrupted. For example, after taking an alpha-1 blocker to treat hypertension. As I mentioned before, like the coronary circulation, autoregulation is an important mechanism in the cerebral circulation, which maintains a constant blood flow over a wide range of systemic blood pressures, from around 60 to 160 millimeters of mercury. I will cover this in detail a bit later. It has been known for a long time that local blood flow in the brain is closely linked to local metabolism, so that blood flow is directed to areas of the brain that are most metabolically active. Recent studies have started to reveal interesting mechanisms that contribute to this local control. This slide shows an image of a small cerebral arteriole in situ and a nearby astrocyte. These structures are clearly linked physically, with the astrocytes sending out projections with end feet that innervate the wall of the arteriole. It is now thought that increased neuronal activity is detected by astrocytes, which signal directly to the muscle cells of the arteriole wall and stimulate the artery to dilate. There is evidence for parallels in the mechanism here to the endothelium-dependent dilation seen in systemic arteries. But research is at an early stage and the precise mechanisms have yet to be agreed on. It has been the close link between neuronal activity, cell metabolism, and local blood flow in the brain is exploited by in vivo studies aimed at understanding brain function, both in animals and humans. These studies use functional imaging techniques, which include PET scanning, or positron emission tomography, and functional MRI, or magnetic resonance imaging. The images collected are assumed to represent neuronal activity in different areas of the brain when subjects are stimulated in particular ways. The example on this slide was used to correlate brain activity with human responses to being presented with a photograph of either a face or a house. But what these images actually measure is blood flow. The interpretation of the data relies absolutely on blood flow and neuronal activity being coupled. PET scanning employs a positron-emitting radioactive isotope, fluorine-18, which is incorporated into the glucose analogue 2-deoxy. PET scanning employs a positron-emitting radioactive isotope, fluorine-18, which is incorporated into the glucose analogue 2-deoxy-fluoro-D-glucose. The glucose analogue is injected systemically and distributes in blood throughout the body. An increase in the concentration of the isotope is detected in areas of the brain where the cerebral vessels dilate and blood flow increases. In interpreting the data, the dilation of the arteries is assumed to directly reflect an increase in brain activity in that area. Functional MRI can be done without injecting any labelled probe, but the underlying principle is the same. It is less invasive because the measurement of blood flow exploits a difference in the magnetic susceptibility between oxyhemoglobin and deoxyhemoglobin. It assumes that I briefly introduced autoregulation earlier. It is a feature of the coronary, cerebral, and renal circulations. Autoregulation is a mechanism that is intrinsic to these blood vessels. It is a response to changes in the environment which acts to maintain a constant blood flow in the face of changing blood pressure. It is illustrated by the graph on this slide. The top trace records flow through an arteriole starting at 5 ml per minute per 100 grams of tissue, when the pressure on the vessel walls was 100 millimeters of mercury. At time 0, the pressure was dropped to 70 millimeters of mercury. The black trace in the upper panel shows the response in the absence of any autoregulation. In this case, there was a passive loss of flow when the pressure fell. The red trace shows what happens when the autoregulatory mechanism was present. Flow initially decreased passively, but the vessel then actively dilated to enable the blood flow to return towards the levels seen at the higher pressure. We know of 3 mechanisms that contribute to this response in vivo. I have mentioned 2 of these before. One involves detection by the endothelial cells of shear stress caused by the blood flowing through the vessel. This triggers the release of relaxing factors from the endothelium, which evoke vasodilation. Another mechanism involves the effects of local metabolites on vascular smooth muscle. The third mechanism is known as the myogenic response. In this case, the smooth muscle cells in the vessel wall contract in direct response to a rise in blood pressure. The response is only seen in the smallest arterioles. Large arteries, even in the coronary, cerebral, and renal circulations, lack the ability to respond to increased blood pressure in this way. This slide shows the results of a study that appears to explain the molecular mechanisms that give rise to the myogenic response. The mechanisms are not fully understood, however, as studies continue to suggest additional and/or alternative mechanisms. In the study shown here, the myogenic response was measured in vessels expressing normal levels of the TRPC6 cation channel subunit. The response was then measured in vessels where expression of the TRPC6 subunit was reduced by treating the vessel with antisense oligonucleotides directed against the subunit. A sense oligonucleotide was used as control. When the normal vessels were exposed to increasing pressures, instead of the vessels getting passively wider, they constricted, showing the induction of myogenic tone. This is shown in the left-hand panel by the blue trace, which should be compared with the black trace, showing the predicted diameter if only passive changes took place. When TRPC6 was knocked down, only passive changes in diameter took place when the pressure was increased. This is seen in the right-hand panel. Similar studies have also shown the involvement of the mechanosensitive cation channel TRPM4. So based on these studies, and a number of earlier studies showing that the contraction also requires calcium influx through voltage-gated calcium channels, the mechanism that is most favoured at the moment for explaining myogenic tone is as follows. TRPM4 and/or TRPC6 sense stretch of the smooth muscle cell membrane due to the rise in pressure. When open, these cation channels allow sodium influx, which depolarizes the smooth muscle cell. This in turn opens voltage-gated L-type calcium channels. Calcium consequently flows into the cell, activates calmodulin, and consequently causes the muscle cell to contract. This slide shows the results of a study that appears to explain the molecular mechanisms that give rise to the myogenic response. The circulation of the lungs differs from the rest of the circulation in that the arteries supplying the lung carry deoxygenated blood from the heart, and the veins return oxygenated blood to the heart. This reflects the primary function of the pulmonary circulation, which is to deliver blood to the alveoli for gas exchange and to return freshly oxygenated blood to all the tissues of the body. As the vessels enter the lung, they branch immediately and very extensively. The walls of the arteries have less muscle than systemic arteries. This is because the arteries operate at a much lower pressure. The mean arterial pressure in the lungs is only around 15 millimeters of mercury, which compares to a mean pressure of about 90 millimeters of mercury in the systemic circulation. The low pressure is functionally important because the lung has to accommodate the entire output of the right ventricle, and the blood is delivered to a very delicate alveolar surface. At high pressure, the capillary alveoli interface would be at risk of damage. In conditions that cause the pulmonary arterial pressure to rise abnormally, the raised pressure results in pulmonary edema due to leakage of fluid from the capillaries. It can also damage the delicate capillaries, leading to hemorrhage. Because the pulmonary vessels are highly distensible and the pressure is low, blood flow within the lung is sensitive to gravity. When standing upright, 3 distinct areas of variable blood flow can be identified in the lung. At the bottom of the lung, the pressure of the blood flowing through the arteries is higher than the air pressure in the alveoli. This favours blood flow from the arteries, and, as a consequence, blood perfuses the capillaries and flows back to the veins. In this area, there is therefore effective delivery of blood to the capillaries for gas exchange. At the top of the lung, the pressure of blood in the arteries is lower than the pressure in the alveoli, which inhibits blood flow, so that the capillaries are not perfused. In the middle of the lung, capillary blood flow is variable because the intravascular and alveolar pressures are close. As you move about, the relative pressures in the vessels and alveoli change, so perfusion of the capillaries will vary. For example, when lying down, blood flow will be more evenly distributed throughout the lung. The regulation of blood flow in the lung employs similar mechanisms to the systemic circulation in terms of regulation by the autonomic nervous system. Reflex regulation of systemic blood pressure involving baroreceptors and chemoreceptors affects the pulmonary circulation in the same way. So a rise in systemic pressure may be mirrored by a rise in pulmonary blood pressure. Humoral control of pulmonary pressure is also similar to the systemic circulation. The endothelium releases similar factors that affect vascular muscle cells in a similar way. A major way in which the pulmonary circulation differs from the rest of the circulation is in its response to hypoxia, which is a major physiological regulator of pulmonary blood flow. Hypoxia causes pulmonary arteries to constrict in a mechanism known as hypoxic pulmonary vasoconstriction. This means that in parts of the lung where oxygen levels are low, the arteries constrict and reduce blood flow to that area. This means that blood is diverted to areas of the lung where oxygen is higher, ensuring the blood is diverted to areas where there is maximum oxygen available for gas exchange. It therefore helps in the matching of blood perfusion to the ventilation of the airways. In the systemic circulation, hypoxia has the opposite effect. In that case, hypoxia signals high activity in the tissue and hence an increased need for oxygen. By dilating vessels in these conditions, hypoxia increases blood flow to the tissue to deliver an increased supply of oxygen. A downside of the constrictor response of the pulmonary circulation to hypoxia is that if hypoxia is global, for example, as might happen at high altitude, or if the mean chronic obstructive arterial pressure rises all the 25 millimeters, and that causes a rise in the pulmonary arterial pressure throughout the lung, hypertension, and this can actually impede oxygen uptake. If the mean a major way in which the pulmonary circulation rises above from the rest of the circulation of mercury is in its response to hypoxia, that is known as pulmonary, which is a major physiological regulator of pulmonary blood flow. Hypoxia causes pulmonary arteries to constrict. This slide illustrates some of the many mechanisms that have been proposed to explain hypoxic pulmonary vasoconstriction. Over the years, it has been proposed to involve a factor released from endothelial cells or from other cell types in the lung. It is generally agreed now that hypoxia directly affects the smooth muscle cells, and probably through an action on the mitochondria. Hypoxia has also been found to inhibit a number of distinct potassium channels, to stimulate calcium channels, and to promote the release of calcium from the sarcoplasmic reticulums. These effects may all be secondary to altered mitochondrial factors, but many of the proposed mechanisms are controversial. For that reason, I do not expect you to remember all of the mechanisms illustrated here. The last specialised circulation we will explore is the fetal circulation. The fundamental difference between the fetal and adult circulations is the location of gas exchange. The vascular structures in the fetus are adapted to extract oxygen as well as nutrients from the placenta of the mother instead of the lungs. So we will start at the placenta and follow the blood around the fetal circulation. From the placenta, the oxygen and nutrient-enriched blood travels in the umbilical vein to the liver. Some of the blood flows through the liver, while the rest passes through the ductus venosus, which is a vascular channel that connects the umbilical vein with the inferior vena cava. The blood that bypasses the liver combines with the venous blood from the liver in the inferior vena cava, from where the blood travels directly to the heart, entering at at the right atrium. In the fetus, there is a direct link between the right atrium and the left atrium, formed by the foramen ovale. This is a small, flap-like structure that forms a hole between the walls of the two atria. Most of the blood flows from the right to the left atrium and then into the left ventricle via the mitral valve. From there it empties into the ascending aorta, then the descending aorta, as would occur in the adult. This takes the oxygen and nutrient-rich blood out to the respiring tissues. The carbon dioxide-rich and nutrient-poor venous blood returns to the right atrium of the heart via the superior vena cava. Note that in the right atrium there is some mixing of the oxygen-rich blood from the placenta with the venous blood of the fetus. Instead of passing to the left atrium, some of the blood in the right atrium enters the right ventricle and is pumped to the pulmonary artery. In an adult, right ventricular blood would be expelled to the lungs for gas exchange. But the lungs of the fetus do not have an air supply and are unable to serve this function. So, although some of the blood entering the pulmonary artery does circulate around the lung to provide oxygen to support tissue respiration, most of the blood leaves the pulmonary artery before it enters the lung to flow directly into the systemic circulation. This is enabled by a structure known as the ductus arteriosus, which is essentially a short blood vessel that connects the pulmonary artery directly to the top of the descending aorta. The reason that a large proportion of the blood in the pulmonary artery is diverted via this route is that, unlike in the adult, the pressure in the pulmonary circulation of the fetus is high, pushing blood away from the lung and into the aorta. Notice that the blood returning to the placenta for replenishment travels from the arterial side of the fetal circulation through two umbilical arteries. This situation resembles the pulmonary circulation, where the arteries carry deoxygenated blood and the vein the oxygenated blood. In that way, it is opposite to every other circulatory system. The fetal circulation is plastic, and at birth, when the baby takes its first breath, things change in order to equip the baby for life outside the womb. The first thing that happens is that the placenta is removed from the circulation as soon as the umbilical cord is clamped. Blood stops flowing through the umbilical vein to the liver, so the blood arriving at the right atrium is now composed entirely of the deoxygenated venous blood from the baby's organs. The oxygen content of the blood entering the right ventricle and then the pulmonary artery is therefore reduced. If the lungs do not quickly start supplying oxygen, the baby becomes asphyxiated. At the same time, the loss of the mother's circulation and exit from the womb also leads to cooling of the blood in the baby. These two factors are sensed by the respiratory centre in the brain, which stimulates the baby to breathe. Over the next few days, the ductus venosus gradually closes to completely block off the umbilical vein. As breathing begins, air gradually replaces the fluid in the alveoli, which is pushed out into the interstitial space from where it enters the capillaries. Air filling the alveoli brings oxygen into the lungs. The oxygen is sensed by the pulmonary arteries. In the same way that hypoxia constricts the pulmonary arteries, at this time the increased oxygen levels dilate them. This reduces the pulmonary arterial resistance and increases the flow of deoxygenated blood to the lungs. This happens simultaneously at millions of alveoli throughout the lung, resulting in a large reduction in resistance and therefore a reduction in pulmonary arterial pressure. This has a knock-on effect on the right side of the heart from where the blood is flowing, causing the pressure in the right ventricle and atrium to fall. At the same time, the increased blood flow in the lung, along with the arrival of oxygen in the alveoli, means that a greater volume of now oxygenated blood is delivered to the left atrium. Thus the pressure in the left side of the heart increases, while the pressure in the right side falls. This has the effect of closing the flap of the foramen ovale, stopping the flow of blood from the right to the left atrium. This happens in the first few minutes of birth. The ductus arteriosus is also able to detect changes in blood oxygen. In this case, like systemic arteries, the ductus arteriosus constricts as oxygen in the blood flowing through it rises. This closes the vessel so that all the blood from the right ventricle now flows through the pulmonary artery to the lungs. This occurs over a few hours, with the ductus eventually being closed completely. Blood stops flowing down the clamped umbilical arteries because there is nowhere to go. The high pressure means that blood flows down alternative branches where the pressure is lower. In addition, the higher oxygen in the blood reaching the umbilical arteries causes them to constrict, so that they gradually shut off completely. Blood stops flowing through the umbilical vein to the liver, so the blood arriving at the right atrium is now composed entirely of the deoxygenated venous blood from the baby's organs. The oxygen content of the blood entering the right ventricle and then the pulmonary artery is therefore reduced. This slide summarises the events described on the previous slide. Please see Blackboard for recommended further reading, as well as links to online resources.